Good afternoon. Today's Tuesday, October 6th, and we are going to be taking a look at the Population Estimation Lab activity regarding two methods, one called market recapture and the other called Quadrat. We began our population studies. We looked at ways that you can assess populations. So we looked at things like population density. We looked at population distribution, which include uniform, random, clumped distributions. We explored the idea of density dependent and density independent factors. We talked about natural ways populations can increase or decrease. Populations can naturally increase through birth rates or immigration with an I, or they can naturally decrease through death rates or emigration with an E. We looked at exponential growth and logistical growth. So we learned a lot about how you measure a population. Once we learned about measuring a population, we applied what we learned to a real population study that's been going on for 50 years in Tanzania, Africa, in a national park called the Serengeti. And in the Serengeti, they focused on the populations of buffaloes and wildebeest. We then shifted over to another part of Africa, into Mozambique, into the national park called Gorongosa, where they did an African elephant population, and ended up actually doing a continent-wide African elephant population. And there we learned about aerial surveys, which were great for grassland savanna elephants. But then we learned about other surveys. We have forest elephants. Forest elephants, aerial surveys don't work. You can't see through the trees. So then they talked about things like acoustic surveys and dung transect surveys. They talked about individual registration, like using radio collars. So we talked about different techniques. Different populations are going to require slightly different methods for estimating population. Because you can either do a total count, which is typically not realistic. You can't just go into Africa, blow the whistle, and all the elephants are going to line up so you can count them. So you typically are doing sampling, and that's where all these techniques come in. So in this activity, we're looking at two sampling methods you commonly would use in a place like Michigan. One is called market recapture. The other is called Quadrat. And so we're going to walk through this together. So let me share my screen. We're going to go to Google Classroom. And so we are talking about this activity right here, Population Estimation Lab, EcPD-8. So let me open that document up. And I would suggest read the introduction. You already should have done the engage activity. That was watching the film about the elephant survey that they did in Africa. And then I would suggest reading the mark and recapture method all the way until you get to the second page where it says mark and recapture formula, and then sort of stop. So you may want to pause the video and take a moment to read those two pages. Go ahead, pause and take a moment to read. Don't just skip over it. So thank you for taking the time to read. So market recapture, it's the idea that we go into an area and we capture a population sample. And that first population sample, we somehow mark it. So you can see in this butterfly, it's been marked with a pink, red, and blue dot, and it's had some numbers written on it with a Sharpie. Doesn't affect the organism, but it allows us to track the organism. Or in this case, we have a snail who's been tracked with a sticker that's yellow with a number on it. And then we release them back into the wild. We give them time to remix with the wild population, and then we go back and do a second sampling. And the whole method is based on how many of the second sample are recaptures, how many of them were captured the first time. So the idea is, if you say catch 20 butterflies and you release them into a population, and say the population's only 50, well, if you have 20 out of 50 butterflies marked the first time, what's the odds of catching marked butterflies again? It's pretty high. So small populations, you're going to have a high recapture rate. But if you put 20 marked butterflies into a population of 2,000 butterflies, the odds of you catching the marked butterflies again is very small. So large populations tend to have a much lower recapture rate. And that's sort of the premise that this method is based on. So here's the formula. And it tells you what each of these variables stands for. So we've got a practice problem here. In this practice problem, it says, let's consider an example. Suppose you want to know how many box turtles are in a wooded park. On the first day, you hunt through the woods and you capture 18 turtles. You place a spot of paint on each turtle's shell and release all turtles back where you found them. So first time we're catching 18 turtles and we're marking them with a paint spot. That's our M, marked individuals released. That is our M value. So then you give enough time for those turtles to go back and mix into the normal population, and you do a second sample. So a week later you return, and with hard work and effort, you catch this time 50 turtles. Of these 50 turtles, 10 are marked and 40 are unmarked. So 10 of them have the paint mark you put there the first time, and 40 of them are new captures. So this represents the S value. The S value is the size of the second sample, which was a total of 50. And the R is the number of marked animals recaptured, which in this case is 10. So we know the M, we know the S, and we know the R. What we're trying to figure out is the N. So we've taken the formula up here. Look where my cursor is. We've taken the formula up here in the middle of the page, and we've rearranged it algebraically into this formula so we can solve for N, because N is the unknown. We want a population size. So in this particular case, we can do the calculations based on the numbers. We need the calculations based on the numbers. So we need M. M was marked individuals that were released, and that was 18. We originally marked 18 turtles. So right here I put 18, because here's this formula up here. 18 is M. S is the size of the second sample, 50. So M times S, 18 times 50, divided by R. R is the number of recaptures. And in our 50 sample the second time, 10 of them had paint on them. They were recaptures. So I plug those numbers into the formula. So I have 18, our M value, times 50, our S value, divided by 10, our R value, and that equals 90 box turtles. That would be our estimated size of turtles for that particular woodlot. So that's an example of how you would use the formula. So let's scroll further, and now there's another practice problem. So a biologist nets 40 leopard frogs from a local pond, tags them with a microchip, and releases them unharmed. A week later, she nets 62 frogs from the same pond, including 26 with tags. Based on this Lincoln-Peterson index, that's the name of the formula, estimate the number of leopard frogs in the pond. So go ahead, take a moment and see if you can figure that out. Go ahead, give the math a shot. You need to know how to use the formula before we start the activity. So I'm going to stop sharing my screen for a second so I can bring up a whiteboard. So here's my whiteboard. And I'm going to come back in about 30 seconds and we'll walk through the problem and see if you got it correct. Okay, that should have been more than enough time to try to figure that out. So let's just walk through and see if we've got it. So again, I want to solve for N, and I can only write so neat with this thing. And so to get N, I have to take M times S divided by R. So if we look at the leopard frog situation, how many frogs were captured the first time and marked? That's our M value. And the correct answer is 48. 48 frogs were caught the very first time and given a microchip. They were marked. Second sample was how big? Well, it tells us in the reading that the second samples were that the second time they caught 62 frogs. And then of those 62, how many of them had tags? We were marked the first time. And it tells us that that is 26. All right? So if we take our calculator, just pull up the calculator here. Where'd you go on me? There it is. All right. And I take my calculator, I take 48 times 62 divided by 26. That's going to estimate our population to be about 114.5. Since we're talking about whole organisms, we're going to round that number either up or down. So somewhere between 114 to 115 frogs is what we would estimate to be in the pond. Right? And so that's what you need to know how to use that formula so we can move forward with the activity. So I'm coming back to the activity. So here's the directions for the activity. I'm going to sort of walk you through it. But basically what we did is we took some, I'm going to stop sharing that just for a moment. We took some basic beans. Here's some kidney beans, red beans. We took some white beans, some navy beans, and we took an empty paper bag. And what they did is they took three normal handfuls of red beans, we call them bean fish, and we put them into the bag. And our very first sample we took was the ones we were going to mark. So my very first sample that I took red beans. I counted how many red beans I got, and I took the red beans and replaced them with white beans. That was how we were going to determine that those were the marked beans. Instead of actually pulling out some paint or something and actually painting the beans, this just makes it quicker. So in the real test, you don't replace anything. You actually have to mark them. But this was a quick, easy way to do it visually. So we took the red beans, we replaced the red beans with the white beans, and that became our M value, okay? So let me go back. So in our particular sample, so here's your data page. First of all, you need to eyeball what you think it looks like. So you can't really see the beans we have. We made a random guess of about 275. You can record these numbers too. And my very first sample that I took and replaced with white beans, I got to check the board real quickly, was 55. So we had 55 individuals we took out of the population the first time and we replaced with white beans. So those are now in the bag. I have the red and the white beans in my bag. And what I have to do for the next 10 trials is shake this up and pull out a sample. All right. Let's move my screen over. So here's my sample, and I'm going to count. There's 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, and total of 29. There's 29. That's my S value. 29 is my S value. We caught 29 beans. And of those 29, how many were recaptured? Those would be the white ones. There was 3. Right? So that is the value that I'm going to put, 29 and 3. And we have to calculate. We'll come back and do that afterwards. So then I took the beans, taking the beans, and I'm putting them back in the bag, and I'm shaking the bag up. Good mix. And I want to be fairly consistent on my sample size. So I'm going to go 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22, 24, 26, 28, 30, 32, 34, 36, 38 total. And of that, 2 of those are recaptures. And I'm going to repeat that 8 more times. I'm going to repeat that 8 more times. Okay? Just making sure I didn't drop any. So I'm going to pause the video for a second and do those other 8 trials, and then we'll come back so that you can get the data. Let me just do a quick pause. Okay, so we have completed gathering our data. So let me go over and share that. So this is the data that you're going to want to record. Those are our samples. So this was the size of our second sample, 10 times, and these were the number of recaptured beans in each of those samples. Now you're going to have to go through and calculate N. Remember, N equals M times S divided by R. So the first one would be 55 times 29 divided by 3. That gives you that answer. Then it'll be 55 times 38 divided by 2. 55 times 47 divided by 6. And you'll get the estimated population size 10 times. You'll then take those 10 numbers, these 10 numbers right here, you'll add them all together, get a total count, and divide that by 10, because you have 10 samples, and that will give you your estimated number. The total, the actual population, then we had to count the beans, and when we counted the beans in the bag, there's actually 411. So we then want to see how close was our estimate to the actual population. So we calculate the percent error. That formula is right here at the bottom of the 3rd page. We take the experimental, which will be this calculated number, subtract the actual of 411, divide it by 411, and multiply by 100. The closer you are to 0, the more accurate your sample is. The further from 0 you are, the less accurate your sample is. So what we saw in class is a lot of groups were all over the place. Some groups were less than 20%. Most of the groups were somewhere between 20 and 50% error, and some groups were over 50% error. And we talked about why would you have this wide range of error if this is supposed to be a good sampling technique? It all has to do with sampling. Different hand sizes, different interpretations of what a quarter handful is. Just our overall sample was small. Most bags only contain 200-300 beans. So we're dealing with really small samples. When we took all the class samples and added them up and did this formula, we got percent error down to about 5%. 5% is acceptable. That means that your estimate is pretty darn accurate to your actual. So if the DNR goes out, they want something that's going to be within about a 5% error. They don't want something that's going to be 30-40% error, because that's not real reliable. So small samples don't work well. You're much more likely to have mistakes, much more likely to have sampling issues, much more likely to have fluke. Bigger samples just simply are stronger. Bigger samples are going to be more accurate. So your percent error may come out pretty large. If it does, it's probably primarily due to the sample size. And if we would take much bigger samples and more frequent samples, we would see that number get tighter and tighter. I hope that makes some sense. You can also then answer these 4 questions. So number 1, we sort of just addressed. More consistent sample size, making sure you mix the bag frequently and well, using larger repeated trials. 2, this will work on any organism that is mobile. Mark and recapture requires the organism to be mobile. So anything that will move and remix with its own population, you can do mark and recapture with. It doesn't work on things that are non-mobile. What are you gonna do? You're interested in studying a particular plant, you can go put a paint spot on the plant, go away for a week and come back, and what, the plant moved? no, the plant didn't move. So populations that don't move, this is not an effective technique. Works quite well for organisms that do move. The last question is about the time between recaptures. It depends on the species you're working with. If you're working with something like, say, mice, you know, well, let's put it this way. If you come back too quick, what's the problem? If you release the organisms the first time and you come back too quickly, what do you run into? Yeah, they may not have had enough time to remix with the population. They may not have had enough time to work their way back through the area. And vice versa, if you come back too late, they could be dead. Somebody could have eaten them, they could have died, they could have emigrated, emigrated and moved somewhere else. So you got to know what you're dealing with. If you're dealing with something like mice, which are bottom of the food chain, you're going to come back relatively soon because those mice may not survive. If you're doing something much more stable, say like wildebeest, that are migratory, then you could have a little bit more time in between. Hopefully that makes some sense. So what do you do if you are interested in doing a stationary population? Say you want to study the population of a particular plant species or a shrub species, or go to the ocean. You want to study coral reefs. Coral reefs don't move. Or you want to study the sponge population at the bottom of the ocean. These use a different method. You can't use mark and recapture. That's where we go into a process called the Quadrat method, which is talked about here. So I suggest pause the video and take a moment to read that first page. Go ahead, read it. Okay, thank you for coming back. So Quadrat method is based on using a quad. Usually the structure is a quad-shaped structure. It's got 4 sides. Quad means 4. So this is going to be our quadrat. It's just 2 straws that have been put together in a square-like structure that are approximately 9 centimeters by 9 centimeters. This is our sampling tool. When we do this outside for real, we use PVC pipe, and we have PVC that's 1 meter by 1 meter. That's pretty standard in the field. And this is going to allow us to do a subsampling of the whole population. So here we're looking at the ocean, and we've got some purple sand dollars and some yellow starfish. Let's say this is 1 meter by 1 meter box. So the overall area is going to be 1 meter by 1, 2, 3 meters. 1 meter by 3 meters. Total area is 3 meters squared. So if the area is 3 meters squared, how many sand dollars are there? In the entire area, there's 1, 2, 3, 4, 5, 6. So the density of the sand dollar for the whole area would be 6 divided by 3, which is 2 sand dollars per meter squared. Starfish, in this case, happen to be the same, 1, 2, 3, 4, 5, 6 in the entire area. Our density for starfish is 6 divided by 3, 2 starfish per meter squared. But by looking at the picture, we can see that it's not equal between quadrats. In this quadrat, on the right, you have way more sand dollars than starfish. So this would be 1 by 1, 1 by 1. And so this would be 4 divided by 1. There's a density of 4 starfish in this quadrat to 1 starfish per meter in this quadrat. But if you go over here, now we've got a density of 3 starfish to 1 sand dollar. So the distribution is a little bit different for the two. So in this case, we're going to be calculating density. We're going to be calculating density. So let's go to our data. And so what we did is we picked one of the squares on the floor. Right, you can see I've got these large square patterns on my floor. Hopefully you can see that. And we picked this blue square. We picked that blue square to be our study area. Now in a real study, you're using something that's miles squared or kilometers squared. Here we're doing centimeters squared. So our quadrat was, that square on the floor is 130 centimeters by 130 centimeters. And so if I do that, 130 centimeters by 130 centimeters, that comes out to 16,900 centimeters squared. And my quadrat is 9 by 9. So 9 times 9 is going to be 81 centimeters squared. And then in class, we changed this to 3. We changed this 3 to 9 so we had more samples. And so 9 quadrats would be 9 times 81. 9 times 81, because each quadrat's 81, and we're using 9 of them. 9 times 81 means we're going to study a total area of 729 centimeters squared. So of that square on the floor, that is 16,900 centimeters squared, we're going to look at, using the quadrat, about 729 of the 16,900 centimeters squared. So it's a very simple technique. These are not mobile organisms. So I'm just going to do this on the tabletop. We did it on the floor, just so you get the idea. So I'm going to see if you can see what I'm doing. I'm taking my beans and I'm pouring them out, and I'm just going to randomly distribute them. Okay, this is smaller than the square on the floor, but this will give you the idea. So they are randomly distributed. They're not going to move. They're not mobile. Let me come out of this view so you can see it even better. Right. So there's my beans spread out. Again, in class, we did it on the floor. And I'm not going to do a total count because normally this would be miles or kilometers big. Way too big for us to do a total count. So instead, I'm going to take my quadrat and I'm going to randomly place it in the environment. And it's going to help me count parts of the environment. Okay, so I'm going to do 9 of these, 9 samples. It has to be done randomly. And I want to, so I might go here and then here and then here and then come down the middle, 1, 2, and 3, and then 1, 2, and 3. I'm going to try to sample as much of the field as I can. And I do not want to sample the same area twice. So I want to sample this and then come back and sample essentially the same ones. You cannot count individuals more than once. So I've got these a lot tighter together than they would be on the floor. But the whole idea is you put the quadrat in there, and then you count the individuals that fall within the quadrat. So I can see there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 red ones, and 1, 2, and 3 white ones. Now in this case, these are 2 different species. We've got a red bean species and we got a white bean species. This has nothing to do with being marked. So we're going to look at the density of the red species versus the density of the white species. So we got the red species and we got the white species. I would then pick this up and I would randomly go to a different area and drop it, and I would make my counts and record those counts. And so I would do that 9 times, trying to cover the whole area. And I'm getting sample numbers without having to count every single individual. Pretty simple technique. Same thing's done underwater if you're doing something with the ocean floor. Quadrat method. So let me go back to the data table. And I'll use data from one of our classes this morning. Okay. All right. So we had these are numbers from one of the groups this morning. So we had a sample of 3 and 1. So in one quadrat, there was 3 red ones, 1 white one. We had another sample of 7 and 2. So I'm putting 3 samples in each, 3 squares were done for each trial. So first time we got 3 and 1, 3 red to 1 white. Second time we got 7 red to 2 white. And the third time, just let me look at my numbers. Third time we got 8 red to 3 white. Then we went and we got 1 and 0. And we got 3 and 0. And we got 10 and 3. And then we got 8 and 1. And we got 11 and 2. And we got 6 and 2. So those were my counts in the 9 quadrats, right? So there was a total of 9 squares that were done. And I'm just going to add these up. So I'm going to do my red ones. 3 plus 7 is 10, 18, 19, 22, 32, 40, 51, 57. So I've got 57 red. Count my white ones, which is the second number. 1, 3, 6, 9, 10. I've got 14 white for a total of 71 total. So those are my bean counts using 9 different quadrats. All right. So now what I want to do is calculate the density. I want to calculate the density. I wish I could write on this screen. It would be a lot easier. Give me a moment. So to get the density for the 9 squares, I'm going to divide these numbers by 729. Divide these numbers by 729. So if I take, this is going to be awkward. Let me try this. Let me try to take a picture of this and see if it'll let me use it. Okay. All right, and I'm going to try to put, I don't know if this is going to work, but we'll find out. Go to the whiteboard. Let me clear this whiteboard. And I don't know if it'll let me put the picture on there or not. It will not. Bloody heck. Okay. So here's our numbers. We've got 57 red. Well, let's do the total first. We've got 71 total in what size area? Well, this 71 total is out of 9 quadrats, which has an area of 729. So we're going to take 71 beans total divided by the area of the 9 quadrats, 729. So if I can get back to that. So 71 divided by 729. Okay. And I apologize, it's not super smooth. I'm just going to go grab my phone for a calculator. So some of you maybe have already done the math instead of waiting for me. 71 divided by 729. That means I'm showing a density of 0.097. We'll go to the nearest thousandth. For total beans, that's total beans. I can also do the density for red beans. And I can also do the density for white beans. So if I go back, red beans was 57. So I would just simply take 57 divided by 729. And that will give me the red bean density. So if I plug that into my calculator, 57 divided by 729, it's going to give me a density of 0.078. And then I can do the same for my white density. I had a total of 14. So I'm going to take 14. Wow, that's terrible. 14 divided by 729. And that'll give me my white bean density. All right, so quadrat method, we're calculating density first, and then we're going to see that we can use the density to then determine the population estimate. And that's going to be 0.019. All right, so total bean density 0.097. red bean density 0.078, and white bean density 0.019. Not coincidentally, the red density combined with the white density gives you the total density. So that's where we're calculating density. So I'm going to go back over here and record my estimated density as 0.097 beans per centimeter squared. Okay. To get the actual, I know the actual number's 411, but to get the density, I have to compare density. The actual density is 411 beans were in that square on the ground, which was 16,900. So to get my actual density, I'm going to take the grand total of the actual count, which was 411, and I'm going to divide it by the area of the entire square on the floor, which was 16,900. And if I plug that in, that's giving me a density of 0.024. Quite a bit smaller than our estimated density. Our estimated density is 0.97, and here it's 0.024. So our estimate is not very good. So if we go to do the percent air using the formula right below, experimental is going to be 0.097. That's the experimental or estimated. Subtract the actual 0.024. Divided by the actual 0.024. Times 100. Oh, and we are way off. That's a 270% error. That's the highest one of all ours. So this would be extremely bad estimating. This would be a terrible example, and we would not want to use this. The problem is our sample is so darn small. Normally we have 8 groups in every hour doing this, and we combine the data from, nearly 30 to 40 samples. When we combine data from 30 to 40 samples, that percent error is nowhere near 270. It's much more like 10% or below. So sample size is a big factor here, but I'm not worried about the percent error really right now, because we're just trying to show you the technique using a simulation. So realize our estimation is not of any value here. It's too far off, just way too far off. But larger, more frequent samples, we could get that percent error down to 10% or less, and we'd be good to go. So here they're asking us to apply what we got. So if you had an area of 3,600 beans based on what we got, how many red beans would you expect? So we have to take our estimated, which was 0.097. Let me clear this. So we had 0.097. I'll just put a B for beans per centimeter squared. And now they want to know how many would that mean if you had an area that was only 3,600 square centimeters. Wow. So the centimeters squared will cancel each other out. And if you take 0.097, 0.097, oh wait, we're doing red beans. I don't want 0.097. Sorry about that. I'm not doing total beans. So I need to change that number to, what was my red bean number? Oh, heck, I just erased it. Yeah. I think it was like, I can't remember now, so I'm just gonna say it's like 0.079, just to show you the technique. So 0.079 times 3,600 would estimate that there would be 284 red beans in a field that was 3,600 kilometers squared. We're assuming, again, every test has an assumption, we're assuming that the bean population is similar to the one in the area that we studied. So we can take our estimated density, multiply it by the area we're studying, and get an estimated total population count. In this case, that's going to be way off because we have such a high percent error. So again, better, larger samples, we could reduce that problem. I hope I'm still making some sense to you. Question 4, you should be able to answer 2 and 3. 1B, very similarly, you're going to use the number for the white beans. Okay. Number 4, quadrat. If I make this quadrat smaller, it's going to give me smaller samples. Therefore, would I have to have more samples or fewer samples? If I'm going to reduce the size of my sample, I'm going to have to increase the number of samples if I want an accurate number. So if you shrink the quadrat, you got to do more sampling. If you make the quadrat bigger, you can do less individual samples. That's sort of the correlation. And then you should be able to figure this page out on your own based on your other work. So I think that'll give you enough to work your way through this. What I'm really most interested in is you understand the basic technique to mark and recapture. I'm not so worried that you can do all the math, but that you understand the basic technique, and that mark and recapture is a very effective population estimate method if it's a mobile population that has a reasonable lifespan. Can't do this with fish flies. Fish flies are mobile, but the problem is what? Yeah, they live less than 24 hours. So that's also going to be limited. But understand, mark and capture, great technique if you have a mobile population that you can somehow mark. And if your samples are frequent enough and large enough, you'll get good results. Understand, quadrat is a way to sample populations that are very non-mobile and tend to stay in one location for long periods of time. Plants, corals, things of that sort. This technique works very well. And again, provided you have enough samples, you can get a very accurate estimate of the population. So those are 2 additional tools in our belt. This is a simulation. So eventually, sometime in the year, we are going to actually go put this to use in the real field and see how well it helps us learn about our own environment. So you need to finish this up and submit this in the next couple days, please. Thanks for staying with me. Sorry that was not the smoothest, but I think it still got the information across. Enjoy your night.